Hi, everyone. Thanks for coming to my talk today. My name is Winston Stauffer. I'm a doctoral candidate from Dr. Chris Gombotsky's lab at SDSU. The title of my talk today is Novel Roles for ATF6 Alpha in the Activation and Differentiation of Cardiac Non-Myocytes. Let's get started. Heart failure occurs when the heart can no longer pump enough blood to meet the needs of the body. It is one of the most widespread causes of death in the United States. 2% of Americans are currently diagnosed with this disease. It accounts for 1 in 8 deaths and $30.7 billion per year in healthcare spending. Heart failure is a common outcome for patients with a history of myocardial infarction, also known as a heart attack. Myocardial infarction, or MI, occurs when a coronary artery that normally supplies blood to the heart becomes blocked, and everything downstream of that dies. It becomes a myocardial infarction. This necessitates that remote areas of the myocardium must work harder to compensate, and cardiac remodeling results, including cardiac myocyte hypertrophy and fibrosis. Eventually, however, cardiac myocyte death and dropout lead to a dilated ventricle that no longer works as a pump. The endoplasmic reticulum stress response is a feature of numerous diseases, including MI and heart failure. The ER is the site of 35% of all translation in the cell. Newly translated proteins move into the ER lumen, where they're folded, and then down the Golgi pathway, where they are secreted or move into cell membranes or organelle membranes in various capacities. Ischemic heart disease, as well as other diseases, feature conditions that cause protein misfolding in the ER, whether that be ATP deficits or calcium imbalances or redox changes. The ER unfolded protein response, or UPR, also known as the ER stress response, senses misfolding and upregulates genes that address this problem. All eukaryotic cells feature an ER UPR. The ER stress response consists of 3 branches represented by 3 intermembrane proteins that act as sensors for ER protein misfolding. All 3 respond to this misfolding by initiating downstream signaling. PERK by phosphorylating EIF2 alpha, which initiates a global decrease in translation. IRE1 splices out a section of XBP1 mRNA, which allows an active transcription factor to be translated. And ATF6 alpha, itself an active transcription factor, travels to the Golgi where it is cleaved. ATF6 alpha has been extensively characterized by the Gombotsky lab as adaptive in heart disease and in other tissues. Under optimal protein folding conditions in the ER, ATF6 exists as an oligomer in the ER membrane. Under conditions that impair ER function, proteins misfold and build up in the ER lumen. ATF6 senses this and translocates to the Golgi, where it is cleaved by Golgi-resident serine proteases. The cleaved cytosolic fragment is an active transcription factor which translocates to the nucleus, where it participates in the upregulation of numerous UPR-related genes. These UPR genes can include protein chaperones, protein disulfide isomerases, or ER-associated degradation machinery, all with the purpose of restoring protein folding in the ER. It can also include non-canonical ATF6 targets that are indirectly related to protein folding in the ER, such as catalase or Rheb. The Gombotsky lab has extensively shown that ATF6 alpha is activated in cardiac myocytes during cardiac disease. This can be caused by protein misfolding, such as that caused by ER stressors like tunicamycin, causing ATF6 to upregulate protein chaperones like GRP78. It can also be caused by oxidative stress, causing the buildup of reactive oxygen species, such as those found during reperfusion injury, which leads to the upregulation of antioxidants like catalase. It can also respond to growth stimuli like phenylephrine, causing it to upregulate the small GTPase Rheb, which induces cell growth via mTORC1. However, none of these processes have been characterized in cardiac non-myocytes. Cardiac myocytes do make up the bulk of the mass of the heart, but are actually in a minority in the cells of the heart. Cardiac non-myocytes include endothelial cells, fibroblasts, immune cells, and mesenchymal cells. They are all essential to cardiac function, but they are often overlooked in cardiac research because cardiac myocytes are more directly related to cardiac function. However, all of these cell types play roles in cardiac injury. This is our rationale in examining ATF6 alpha in the context of cardiac non-myocytes. We know that ATF6 alpha is important in preserving heart function during heart disease, and it's only been specifically investigated in cardiac myocytes. But we know that cardiac non-myocytes also play critical roles in these diseases, so we therefore hypothesize that ATF6-alpha may also be important in the function of cardiac non-myocytes and in previously unexplored cardiac disease models that involve the activity of non-myocytes. Our aims were as follows. We investigated ATF6-alpha in an in vivo murine injury model that involves cardiac non-myocytes and had not been previously explored in the context of ATF6. This was an MI-induced model of heart failure. We also investigated two isolated murine cardiac non-myocyte models, c-KIT-positive murine cardiac stem cells, or CSCs, and adult murine ventricular fibroblasts, or AMVF. Aim 1 was to determine the role of ATF6-alpha in MI-induced heart failure. Our model was an MI heart failure surgery in black 6 C57 mice. This was achieved via permanent occlusion of the left anterior descending artery, or LAD. Also known as the widowmaker, this artery is the most commonly associated with human disease. The mice were allowed to progress to heart failure for 11 weeks, and they were subjected to biweekly echocardiograms to examine function starting at week 1. The mice were then taken down for infarct size after 1 week, and then biochemistry for heart failure markers after 11 weeks, as well as heart weight and tibial length measurements. ATF6 was examined by comparing wild-type mice to ATF6 global knockout mice that had had ATF6 deleted in all tissues from birth. Starting at 1 week, mice were subjected to echocardiography. Sedated mice were strapped to a heating table and an ultrasound probe positioned in such a way as to give a short-axis view of the heart looking down the center of the left ventricle. The short-axis view allows us to watch heartbeats in real time, and the M mode, or line scan mode, allows us to examine the relationship in the internal diameter of the left ventricle from systole, or contraction, to diastole, or relaxation. This relationship allows us to calculate various parameters of cardiac function, including fractional shortening or ejection fraction. As is typical of post-MI mice with large infarcts, the left ventricular diameter can become dilated, the walls can become thin, and there is little difference between diastole and systole, indicating a decrease in function. A subset of the mice were also taken down and their hearts removed and dissected into 1 millimeter sections with a tissue matrix and subjected to TTC staining, which stains viable tissue, which was not infarcted at the time that mouse was taken down, stains it red or pink, whereas infarcted tissue remains pale. You can also so you can see the difference between normal and post-MI hearts. You can also tell the difference by the internal diameter being small in the normal mice and much more dilated in the post-MI mice. We hypothesized that after 1 week, the ATF6 knockout mice might perform worse than their wild-type counterparts after MI. However, echoes showed no difference in heart function, as measured by the ejection fraction, the percent of blood ejected from the heart with each beat, though we did see a decrease for both groups post-MI as expected. Additionally, TTC staining showed no significant difference in infarct size between wild-type and knockout mice. This was not entirely unexpected because of the nature of permanent occlusion surgeries means that there is no chance for any of the downstream tissue to recover, so it would make sense that infarct size might be similar, regardless of the presence of ATF6 or not. We then continued with the 11-week time course with the rest of the mice. These mice were taken down for biochemistry so that we can run qPCR looking for the induction of fetal genes. These fetal genes are certain genes that are expressed in the fetal heart that are not normally expressed in the adult heart. This likely in part reflects differences in energy metabolism, where the fetal heart uses carbohydrate substrates and the postnatal heart switches to fatty acid oxidation. The failing adult heart, however, returns to the expression of these fetal genes. This includes a switch to fetal isoforms of contractile proteins and the expression of natriuretic genes in the ventricles. The expression of these genes is thus an indicator of heart failure. Additionally, as mentioned before, these mice also got biweekly echocardiograms and were also measured for heart weight relative to tibial length at their endpoint. qPCR results after 11 weeks showed that ATF6 knockout post-MI hearts, shown here in dark orange, had undergone a dramatic shift from the adult alpha myosin heavy chain isoform to the fetal beta myosin heavy chain isoform. The resulting ratio of beta to alpha is a leading indicator of heart failure. Note that this shift is much more significant for ATF6 knockout post-MI hearts than for their wild-type counterparts. Additionally, transcript from natriuretic genes ANP and BNP were also elevated in ATF6 post-MI hearts. Neither gene is highly expressed in the adult ventricular myocardium unless as part of the fetal gene program during heart failure. Again, this shift was highly significant for ATF6 knockout post-MI hearts, but not for wild-type counterparts. Biweekly echocardiography on the same mice showed that ATF6 knockout post-MI hearts decreased in function faster than wild-type, as measured by ejection fraction. Additionally, systolic left ventricular anterior diameter, a measure of ventricular dilation, was also greater in the knockout hearts at 2 time points. However, there was no observed difference in heart weight, indicating no increase in hypertrophy in the knockout hearts. Our AIM-1 conclusions were that without ATF6 alpha, mouse hearts declined in function faster after MI as measured by ejection fraction. Additionally, fetal gene expression was greatly increased. This despite no difference in infarct size. This may indicate that ATF6 alpha can't stave tissue downstream from the permanent occlusion, but does help remote regions of the heart to compensate. In AIM-2, we investigated the role of ATF6 alpha in c-KIT-positive cardiac stem cells. These were isolated from the adult mouse heart by magnetically sorting based on the c-KIT surface marker. They have been shown to differentiate in response to cardiac injury, though mostly into endothelial cells, and rarely, as was earlier suggested, into cardiac myocytes. Based on this earlier data, they had been used in stem cell therapy trials of MI-induced heart failure patients. While these trials produced some positive, albeit ambiguous results, these positive results may have been more due to secretion of paracrine factors than, as was believed at the time, differentiation into new cardiac myocytes. Nevertheless, these cells drew our interest as a dynamic cardiac non-myocyte capable of differentiation. To manipulate ATF6 in these cells, we used siRNA and chemical inhibitors for loss of function, as well as a chemical activator for gain of function. Multiple maneuvers were performed in these cells to affect loss of function and gain of function of ATF6. As discussed before, under control condition, ATF6 exists as an oligomer in the ER membrane. Upon ER stress, it monomerizes and translocates to the Golgi, where it's cleaved by serine proteases, and the cytosolic fragment, which is an active transcription factor, moves to the nucleus to activate its gene program. Loss of function was affected in one case with knockdown using siRNA targeted to ATF6 transcript. Another method was to treat with a chemical inhibitor called PF429242, which is a serine protease inhibitor. This means that while ATF6 can be activated by ER stress and translocate to the Golgi, it cannot be cleaved by the serine proteases normally present there. Thus, the ATF6 transcription factor is not liberated from the Golgi and cannot proceed to the nucleus to affect its gene program. For ATF6 gain of function, a chemical activator called compound 147 was used. 147 acts by converting oligomeric ATF6 to monomeric, and thus promotes its translocation to the Golgi without requiring additional ER stress or protein misfolding in the ER. Thus, the effects of endogenous ATF6 can be examined without the confounding effects of the other branches of the ER stress response. One way to measure the differentiation status of cardiac stem cells is to measure the induction of transcript of differentiation markers towards various cardiac lineages. Treatment with the glucocorticoid dexamethasone can be used to induce differentiation markers in stem cells of various types. qPCR in cardiac stem cells treated with dexamethasone over a 7-day time course show that dexamethasone treatment in purple induces the differentiation markers alpha smooth muscle actin for smooth muscle lineage and GATA4 for early cardiac lineage. To determine the effect of ATF6 on CSC differentiation, we first examined these same lineage markers via qPCR in CSCs with ATF6 alpha loss of function with and without dexamethasone treatment. As expected in the purple column, dexamethasone greatly induced the alpha smooth muscle actin transcript. However, this induction in the striped column with siATF6 on top of it was greatly enhanced. The same was true when chemically inhibiting ATF6 with serine protease inhibitor PF429242, which also induced cardiac marker GATA4. demonstrating that ATF6 loss of function induces some of these differentiation markers, especially alpha smooth muscle actin. Meanwhile, ATF6 activation by the compound 147 decreased smooth muscle actin expression, shown here in pink, and completely blunted induction by dexamethasone in the striped column, though it did not have an effect on GATA4, suggesting that ATF6 alpha gain of function reduces the differentiation marker alpha smooth muscle actin. Because our discovery of the effect of ATF6 on cardiac stem cell differentiation influenced so much of the direction of AIM-3, I will elect to conclude AIM-2 here. To summarize some of our other findings, however, CSCs were also found to be more sensitive to ER stress than cardiac myocytes. In the case of dapsogardin, an ER stressor which depletes ER calcium levels, CSCs were found to be sensitive to dapsogardin doses that were orders of magnitude less than those required to affect myocytes. Furthermore, ATF6 was found to be necessary for cardiac stem cell proliferation even in the absence of overt ER stress. We subsequently examined the effect of ATF6 loss of function on numerous cell cycle genes and oxidative stress genes in cardiac stem cells. However, it was our interest in the effect of ATF6 on the maintenance of CSC stemness which interested us most, and especially its effect on alpha smooth muscle actin. The reason is that alpha smooth muscle actin is a marker for another well-known cardiac non-myocyte, in this case cardiac fibroblasts. Cardiac fibroblasts exist in a quiescent state in the myocardium until activated by a signal such as TGF-beta or injury such as myocardial infarction, pressure overload, or inflammation. Activated cardiac fibroblasts undergo a transdifferentiation process into a new cell type called myofibroblasts. Myofibroblasts are characterized by the increased production of extracellular matrix proteins, primarily collagen, and the induction of alpha smooth muscle actin contractile proteins. Myofibroblasts are required to activate, migrate to the wound area, secrete collagen or ECM fibrotic material, and through the induction of alpha smooth muscle actin contractile proteins, begin to contract like a smooth muscle cell in order to close the wound. Because of the effect that ATF6 had in blunting smooth muscle actin induction in cardiac stem cells, we wondered what its effect would be in cardiac fibroblasts. And so, in AIM-3, we aimed to investigate the role of ATF6 in cardiac fibroblasts. Our model were primary murine cardiac fibroblasts that were isolated from adult mouse hearts, specifically adult mouse ventricles, and so we call them AMVF for adult mouse ventricular fibroblasts. We also conducted many experiments in NIH3T3 fibroblasts, which are an immortalized cell line, in order to check for the specificity of the role of ATF6 in cardiac fibroblasts. We also examined fibroblast markers and fibrosis in permanent occlusion MI surgery that was identical to that used in AIM-1. We also used transaortic constriction surgery, which induces pressure overload, simulating high blood pressure, and induce cardiac hypertrophy and eventual failure. Fibrosis is also a major feature of this injury model as well. Our ATF6 maneuvers were in ATF6 knockout mice, siRNA for a loss of function, and again for gain of function, the chemical activator 147. In the context of MI, cardiac fibroblasts, as stated earlier, exist in the uninjured heart amongst the cardiac myocytes. And then after MI, when this area in gray here has become infarcted, has all these dead cardiac myocytes, the activated cardiac fibroblasts are migrating to this region, are in and amongst the dead myocytes, and are secreting this blue ECM, mostly collagen fibrotic material. Now, 7 days after MI, the cardiac myofibroblasts are fully activated and the scar is becoming more mature. And so now that even though this area is no longer contractile, it is at least stable and is no longer in danger of rupture. Whether this is a good thing long term is debatable because, again, you don't want the heart to rupture, but fibrotic material is typically stiff and unyielding, doesn't contract, and so it actually ends up being a hindrance to the overall function of the heart long term. and overactive fibrosis is a common feature of many forms of heart failure. When we were first looking into fibrosis in the heart in the context of various injury models, we used samples that we had previously generated from other studies, such as AIM-3 of this dissertation, to examine fibrosis markers in these other injury models. So for example, in this MI model from AIM-1, we took RNA from these hearts and ran qPCR for fibrosis genes, by which I really mean markers of myofibroblast activation. And we found that alpha-smooth-muscle-actin and collagen 1A1, both markers of myofibroblasts, were elevated in these hearts. The caveat here is that these are ATF6 global knockout mice, meaning that ATF6 is deleted in all of the cells of the heart as well as all the tissues of the body. So these are not necessarily coming from just the cardiac fibroblasts, but it is an indication that there is elevated, perhaps fibrotic activity going on in these hearts. So what we did was we took additional hearts that had been subjected to the same surgery, but had been embedded in paraffin blocks, sectioned them, put the sections on glass slides, and then stained them with picoserous red, which is a fibrosis stain primarily of collagen. And under the microscope, you get these nice images where the pink or red area is the fibrotic material, the pale area is not fibrotic, and this is a zoom in of this section here, so you can get a good idea of the area of fibrosis after this MI. So these paraffin-embedded MI hearts were actually generated by Khalid Azizi, a master student from the lab a few years ago. And so these were both stained, both of these conditions were stained with picoserous red. And you can see here that there's a variable amount of the pink area, the fibrotic area, in all these conditions, and it's mostly appears to be dependent on the size of the infarct. So the more infarcted the area, the more fibrotic area you get. We did try to quantify this using this Keyence software, which scores all of this pink area here as green here, and all of the non-fibrotic or pale area up here is down here scored as purple, and then it adds up all of the green area and measures that area and averages it out, and you can get this quantification. Though we did not find a significant difference in the fibrotic area between wild type and ATF6 global knockout, which was surprising given that we had seen a fairly obvious difference in the induction of fibroblast activation markers. We next decided to try an injury model that was less dependent on something like the size of the infarct, and went with a transaortic constriction surgery, which we also had previously existing samples for. Transaortic constriction, or TAC surgery, involves the constriction of the transverse aorta between their left and right carotid arteries. This simulates hypertension because with each beat, the left ventricle pumps blood into the aorta. The aorta usually expands, but now that it's constricted, it can't, and provides a lot of pressure back into the left ventricle, causing it to have to work harder, similar to systemic hypertension. Over time, this can lead to pathological cardiac hypertrophy. Let's see this, before TAC is much smaller than the myocardium in the after TAC. Eventually, this features also cardiac myocyte death, dropout, and thus features a lot of interstitial fibrosis. So with the TAC hearts we had on hand in paraffin blocks, we did the same thing, sectioned them on the microtome, stained them in picoserous red, examined them under the microscope. You can see an example of the result here with the fibrotic area in dark pink and the non-fibrotic areas in pale. These samples were from surgeries performed by Eric Blackwood, another PhD student in the lab, now graduated. And what's apparent from picoserous red staining is so there's no increase in fibrosis in the sham hearts, a little bit of an increase in the wild type TAC hearts, but much more of an increase in the ATF6 global knockout TAC hearts. This can again be visualized using the Keyent software, where green represents the area stained by picoserious red, the fibrotic area, and you can see how much more of it is in the global ATF6 knockout heart. And it can be quantified here. So in orange, again, you can see much more of the fibrosis in the global ATF6 knockout heart compared to wild type. Now, the caveat here is that, of course, this is an ATF6 global knockout, so we don't know exactly in what cell type the lack of ATF6 is causing an increase in fibrosis. The obvious way to address this is, of course, to use a cell-specific ATF6 knockout. And while we don't have a fibroblast-specific knockout, we do have a model where ATF6 is knocked out only in the cardiac myocytes of the heart. These samples were generated from mice that had undergone the same TAC surgery as before, but now we're comparing wild-type mice to ATF6 cardiac myocyte-specific knockout, or ATF6 CKO. This was achieved by expressing a Cre recombinase behind a cardiac myocyte-specific promoter, in this case cardiac troponin T, and a background of ATF6 floxed mice to achieve cardiac myocyte-specific ATF6 knockout. As before, when we stain with picoserious red for fibrosis, we don't see any increase in fibrosis in the sham hearts as expected. We do see an increase in the wild-type TAC hearts, and we also see an increase in the ATF6 cardiac myocyte-specific knockout TAC hearts, but to a lesser degree in this case. This is quantified in the Keyent software, where green again indicates the fibrosis, and quantified here where you can see the increase in the TAC hearts for the wild-type mice, as well as the increase in the TAC hearts for the ATF6 alpha cardiac myocyte-specific knockout mice, but now to a lesser degree. So this would indicate that the lack of ATF6 in cardiac myocytes is not the cause of the increase in fibrosis we saw in the previous figure. While it's not as specific as we would like eventually cardiac fibroblast-specific knockout. This does help narrow down our list of choices for the cell types that are responsible for the increase in fibrosis in the heart. To get a better idea of how ATF6 acts in cardiac myocytes, cardiac fibroblasts specifically, we chose to use an isolated adult ventricular fibroblast model in culture. AMVFs were isolated from adult mouse hearts by culturing the non-myocyte fraction from a digested heart under conditions which selected for fibroblast proliferation. As before, wild-type control AMVF have functional ATF6 which can be activated by ER stress to affect its downstream gene program. For ATF6 loss of function, we can isolate AMVF from ATF6 knockout hearts, that would be ATF6 global knockout hearts, which do not have ATF6 in any cell type, including fibroblasts. We can also isolate wild-type fibroblasts and then knock down ATF6 using RNAi. Lastly, again, for ATF6 activation, we can use compound 147 on wild-type fibroblasts, which activate ATF6 without the need for overlying ER stress. To induce differentiation in cardiac fibroblasts, we treated with the TGF-beta canonical fibroblast activator. TGF-beta is held inactive in a latent complex in the extracellular matrix, and when activated, it binds TGF-beta receptor 2, activating its kinase function to complex with and phosphorylate TGF-beta receptor 1, itself a kinase. And then the TGF-beta receptor complex acts to phosphorylate secondary messenger proteins like SMAD2, which then co-localize with co-SMAD, SMAD4, and this complex then moves to the nucleus, where it acts to directly upregulate fibrosis-related genes, such as alpha-smooth muscle-actin and collagen. After confirming that TGF-beta treatment did indeed induce activation markers in our cultured fibroblast model, we treated with TGF-beta in the context of ATF6 loss and gain of function. And what we found was that ATF6 alpha loss of function induced myofibroblast markers with and without TGF-beta. Fibroblasts that had been isolated from ATF6 knockout mice, global knockout mice, already had elevated myofibroblast markers, smooth muscle actin and collagen 1A1, which were further increased with treatment with TGF-beta. So they were both hyperactivated to begin with and could be hyperactivated further with TGF-beta treatment. We found similar induction of smooth muscle actin in wild-type fibroblasts that had ATF6 knockdown via RNA interference. Conversely, ATF6 alpha gain of function acted to reduce myofibroblast markers. In this case, activation was again achieved with compound 147. which decreased smooth muscle actin and collagen 1A1 levels, and also blunted their induction by TGF-beta. So similar to the dynamic observed earlier in cardiac stem cells, in which ATF6 gain of function acted to reduce the induction of differentiation markers in the context of dexamethasone treatment. We wanted to examine the functional consequences of ATF6 blunting myofibroblast activation markers. This would give us an idea of whether the changes we observed at the qPCR level, that is, transcript level, were also having effects at the protein and functional level. One functional assay for fibroblasts is the collagen gel contraction assay. In this assay, fibroblasts are embedded in a collagen gel disc. This disc is polymerized into a semi-solid state, which then is allowed to freely float in the culture media. When treated with TGF-beta, as is normal with fibroblasts, this causes an upregulation of alpha smooth muscle actin protein. To review, the purpose of smooth muscle actin in myofibroblasts is to allow the cell to act as a smooth muscle cell and contract, pulling on the surrounding tissue and closing the wound area. Thus, when treated with TGF-beta, the fibroblasts in the collagen gel disc become activated, turning into myofibroblasts, which then contract, pulling on the surrounding collagen matrix and reducing the diameter of the disc. Thus, the smaller the disc, the more activated the fibroblasts in the culture are. This allows for a simple output for the measuring of fibroblast activation. So here's a picture of the collagen gel discs in the wells floating in growth media. And when we add TGF-beta, the diameter of the gel discs decreases, which is quantified here as fold of max contraction. So higher bar means more contraction, demonstrating the expected effect of TGF-beta on fibroblast contraction. When we activate ATF6 with 147, though, what we found is that the collagen gel discs actually get larger, so less contraction, indicating that there's probably some minimal level of activation happening even in the control cells that's being blunted by the presence of 147 and thus ATF6 activity. And when added in the context of TGF-beta, 147 acts to blunt the effect of TGF-beta on fibroblast contraction. Again, quantified over here. The 147 with TGF-beta is not significantly different from control, demonstrating that TGF-beta can't induce fibroblast contraction in the presence of ATF6 being chemically activated by 147. So this is in agreement with the earlier data where we see decreases in smooth muscle actin transcript with ATF6 activation, that we're actually seeing a functional consequence where the cells are not able to contract nearly as much as when ATF6 is not chemically activated. In order to visualize the effect of ATF6 alpha on cardiac fibroblast activation, we used immunofluorescence microscopy to immunostain alpha smooth muscle actin and AMVF in culture. Cell nuclei were stained in blue with a Hush stain, while smooth muscle actin was stained green with Cy3. As you can see, there are some cells that have green puncta for smooth muscle actin, and others that have these branching networks of fibers. These smooth muscle actin fibers are also called stress fibers and are a very commonly used method for detecting the activation of cardiac fibroblasts and their transition into a myofibroblast phenotype. The number of stress fiber positive cells is quantified in the upper left corner of each image, and the total number is averaged here. So in this case, in the control, we can expect to find about 1.5 stress fiber positive cells per field. This indicates that even under control conditions, as we've seen previously, there's some level of fibroblast activation, probably just as a consequence of being cultured in a dish. When we treat with TGF-beta, as you might expect, we get a dramatic increase in the number of stress fiber positive cells per field, about 4.5 stress fiber positive cells per field here. And though that's what we were quantifying, I think it's also apparent that the intensity and complexity of the stress fiber networks was also increased in the TGF-beta treated fibroblasts. So this is a visualization of why TGF-beta activated myofibroblasts are able to contract as seen in previous sets of experiments. When we activate ATF6 with 147, we see a little bit of a decrease in the number of stress fiber positive cells under control conditions compared to control conditions. But really dramatically, when you treat with 147 and TGF-beta concurrently, we find that ATF6 activation completely blunts the effect of TGF-beta on stress fiber formation. There are some that have very faint stress fiber networks, but they were actually pretty hard to find. And again, the intensity and complexity of the stress fiber networks are also greatly reduced when treated with 147 on top of TGF-beta. So this provides a visualization of why the 147 treated fibroblasts in the previous experiments did not contract as well as with TGF-beta alone. And this now shows us that the effects we saw earlier of ATF6 blunting the induction of smooth muscle actin and collagen transcripts and the blunting of fibroblast contraction is now, we're confirming that visually with the appearance of smooth muscle actin protein and the structure of those fibers. To determine how ATF6 was affecting TGF-beta signaling, we looked at some of the downstream events in the TGF-beta SMAD signaling cascade, specifically the phosphorylation of SMAD2. To review, SMAD proteins are phosphorylated by the TGF-beta receptor complex. They then co-localize with SMAD4, and the complex then moves to the nucleus, where they upregulate fibrosis-related genes like smooth muscle actin. Thus, when we treat fibroblasts with TGF-beta, we can expect to see an increase in phosphorylated SMAD2. Cell extracts from cultured AMVF were separated via SDS page and probed for phospho-SMAD2 on an immunoblot. And you can see in this center band, when you treat with TGF-beta, phospho-SMAD2 greatly increases, quantified here, as expected. However, when we knocked down ATF6, we consistently saw a slight but significant increase in phospho-SMAD2 levels, indicating a measurable increase in the activity of this pathway even without TGF-beta. Conversely, ATF6 activation by 147 treatment caused a slight but again significant decrease in phospho-SMAD2 levels, indicating that ATF6 may be acting by preventing phospho-SMAD2. If SMAD phosphorylation is the reason we see increased myofibroblast activation during ATF6 loss of function, then inhibiting the TGF-beta SMAD pathway, including the phosphorylation of SMADs, should be sufficient to prevent the induction of myofibroblast activation genes like smooth muscle actin that are induced when ATF6 is silenced. The TGF-beta inhibitor, SB431542, acts by preventing the kinase activity of the TGF-beta receptor complex, which prevents the phosphorylation of SMAD2. Thus, we can predict that treatment with SB431542 will prevent the induction of fibrotic genes by TGF-beta treatment. And this is what we see when we look at smooth muscle actin transcript induction by TGF-beta under control conditions, which is blunted by treatment with SB431542 as expected. In cells that have ATF6 knockdown, as before, we see an increase in smooth muscle actin transcript, both without and especially with TGF-beta treatment. However, SB431542, the TGF-beta inhibitor, also inhibits the induction of these genes by ATF6 knockdown. And you can see that the difference between control and ATF6 knockdown, which is significant here under control conditions, is no longer significant if you treat with the TGF-beta inhibitor. This inhibitor should have nothing to do with ATF6, and yet it seems to be acting on ATF6's, or rather it appears to compensate for the absence of ATF6. This is a strong indication that ATF6 does indeed act on the TGF-beta pathway at the level of SMAD2 phosphorylation, the same level as SB431542. Because ATF6 is a transcription factor and is not known to influence the phosphorylation of any proteins directly, we reason that ATF6's effect on the TGF-beta pathway may be the result of genes that it induces as part of its gene program. Recently in the Gomlatsky lab, numerous non-canonical ATF6 genes have been discovered, like catalase or Reb, that influence processes that are at best indirectly related to ER stress and proteostasis in the ER lumen. Using extracts from AMVF treated with 147 for ATF6 activation, we looked at a wide variety of fibrosis-related genes in this PCR array. The green dots are genes that are increased by ATF6 activity, while the red dots are genes which are decreased by ATF6 activity. It should be apparent the vast majority of these genes are downregulated by ATF6 activity. Conversely, when interrogating cell extracts from AMVF with ATF6 knockdown, most of the genes are actually upregulated, as shown in green here. This indicates that ATF6 may be acting as a global inhibitor of fibrotic-related processes, as evidenced by its wide effect on a wide variety of fibrosis-related genes. Notably perturbed genes are listed here, which were especially upregulated with 147 and downregulated with siATF6, or vice versa. Some of these genes, like IL-1 alpha, which are induced by 147 and decreased with siATF6, are known to be inhibitors of the TGF-beta pathway. Other notably downregulated genes include two TGF-beta isoforms, 2 and 3, as well as both TGF-beta receptors, 1 and 2. This array gives a wider view on the broad effects of ATF6 activity on fibrosis. To conclude AIM-3, we propose that ATF6 acts at the level of SP431542 to prevent the phosphorylation of SMAD2. This is likely due to ATF6 acting through its gene program to upregulate a TGF-beta negative regulator. To date, we have interrogated numerous potential candidates, including IL-1 alpha, PMEP1, SMURF1 and 2, BNP, and SRF, as possible candidates for this negative regulator. However, to date, we have not yet found it. To summarize, for AIM-3, we conclude that ATF6 alpha activation suppresses transcript associated with myofibroblast activation in AMBF and also blunts activation by TGF-beta. ATF6 knockout or knockdown induces these same transcripts and enhances their expression by TGF-beta. Furthermore, induction of these same transcripts by loss of ATF6 alpha can be compensated for by treatment with the TGF-beta inhibitor SP431542. ATF6 activity also inhibits functional readouts of myofibroblast activation, as measured by fibroblast contraction, as well as the appearance and formation of alpha-smooth muscle actin stress fibers. Lastly, ATF6 suppresses most gene transcripts in a fibrosis PCR array, while ATF6 loss of function does the opposite. Future directions for this study will therefore focus on continuing to narrow down where exactly ATF6 influences TGF-beta signaling. Does it indeed induce a TGF-beta negative regulator, and if so, which is it? One potential candidate is an miRNA which did not show up in the fibrosis array called miR130B. 130B was found to be heavily upregulated in RNA-seq datasets from mice with ATF6 activation in their hearts. miR130B is also predicted to downregulate TGF-beta receptors 1 and 2, as well as downstream SMAD proteins like 2 and 4. It would be interesting to know if ATF6 activation, in the absence of miR130B, would still be enough to downregulate TGF-beta signaling. Another avenue of investigation might focus on the ATF-CREB transcription factor family member ATF3. ATF3 was recently found to downregulate cardiac fibroblast activation. In that study, this was found to be due to ATF3's downregulation of MAT kinase and subsequent downregulation of downstream p38 signaling. As a part of this study, we did interrogate p38 signaling in the context of ATF6, gain and loss of function in cardiac fibroblasts, but found no change. However, the effect of ATF3 was found to be due to binding to HDAC1 in the nucleus. ATF6 has also been found to bind HDAC1 in the nucleus in such a manner so as to downregulate other signaling pathways, such as SRABP2. Future studies should elucidate whether ATF6 is also binding HDAC1 in the context of cardiac fibroblasts. Another recent study showed that ER-associated degradation machinery, including HERD1, are responsible for ubiquitolating TGF-beta and thus downregulating it. HERD1 negative regulation of TGF-beta was found to be necessary for proper maturation of beta cells. It would be of great interest to know whether TGF-beta ubiquitination levels are changed with or without ATF6, since HERD1 is heavily upregulated by ATF6 activity. It would also be interesting to know whether ATF6 could continue to downregulate TGF-beta signaling in the absence of HERD1, or other ER-associated degradation machinery. As mentioned before, other future directions should include cell-specific knockout models, specifically knockout models featuring cardiac stem cells and or fibroblasts. Numerous cardiac fibroblast markers exist, such as TCF21, the promoter of which could be packaged with Cre recombinase and a viral vector to effect knockout in ATF6 flocks to mice. Investigating fibrosis using the surgical models used in this study with fibroblast-specific knockout mice would be very important in revealing the role of ATF6 in cardiac fibroblasts specifically. Okay, so I'd like to thank my mentor and chair, Dr. Chris Glombotsky. It's a real privilege to have gotten to work in his lab. It's a really great, supportive environment, very collaborative, and I really learned a lot from him over the years. I'd also like to thank my committee members, Dr. Bernstein, Dr. Zayas, Dr. Heller-Brown, and Dr. Niwa, for being so generous with their time and support as part of my committee. At the Glombotsky Lab, as I said, it's a really great place to work and learn. And just to highlight a very few, Adrian and Eric, my fellow PhD students, were great friends to have along the way. Both provided a lot of advice and consultation. Eric, in particular, as I mentioned, supplied the TAC parts used in AIM-3. And then Khalid Azizi, one of our old master students, who was also a great friend, supplied some of the MI paraffin-embedded MI hearts, as well as taught me how to do the MI surgeries myself, as well as most of the takedowns. Shirin was a great mentor to have, especially my first year in the lab. Donna is the Glombotsky Lab expert and teaches all of us pretty much everything we know about research. Ingrid was very generous with her time, helping me learn microscopy and sectioning using the microtome. Andy was also a great friend and collaborator. Monty, Lauren, Tobias, and Alina all helped keep the lab together. Alina especially for running the mouse core. And Haley for helping me with the cardiac stem cell experiments in their initial stages. Outside of the Glombotsky lab, we had a bunch of other SDSU collaborators and staff that I need to thank. Jenny Waters for helping me with the immunocytofluorescence experiments, as well as the writing of my dissertation. Patty and Medora and Renee and Jamie for helping me and all SDSU grad students navigate the program. Gina, Eugenia, and Elisa for helping us with our funding and running the SDSU Heart Foundation. Dr. Foyer for letting me use his lab's microscope and their microtome. Natalie for being a great source of advice and also for letting me endlessly use her paraffin deparaffination station. And I'll also just highlight Larkin and Juliana and the rest of the OLEC staff for doing the work they do every day in the vivarium and helping everybody that uses the mice. Thank you so much to everyone. I'd also like to acknowledge my ARCS Foundation scholarship, my Reese Steely Research Foundation for funding me. And with that, I'll just say thanks to you for watching, and I look forward to hopefully seeing most or all of you at my Zoom meeting on Monday, July 6th at 1:10 p.m. for Q&A. And look for a link in your email forthcoming. It's not out yet, but keep an eye out. And with that, I'll say thanks again, and see you there.